Good morning. There it is. It's great to see everybody today. It sounds like there are some tired friends here because it's Monday morning and a lot of us stayed up all night this weekend jumping around at prom and after prom. So thanks for coming back this morning and being with us. Today we have an exciting opportunity for you to learn together with experts from around the world. But my job today is to introduce Eric Hansen. Eric Hansen is our city manager, and we are so fortunate in Mason to have a partnership with our city that brings in experts from around the world to be a part of our STEAM and our STEM initiatives here at Mason High School and throughout our district. So this is the kickoff to a collaboration of Innovation Week. We have our city council members here and Sinclair and some other partners. So we are very fortunate. So if you would please join me in a warm welcome to our city manager, Eric Hansen.
Thank you, sir. You're on. Thank you. City of Mason is excited to be here, and I'll tell you why. When you spend a lot of time working on something, it's nice to get out of the office, get with some friends and with some partners, and do something outside of the office, outside the classroom maybe. And that's what this is for the City of Mason. We're excited to partner with the schools, with several of our biohealth businesses, to bring what we think will be a very exciting, progressive thing for a city like Mason. And with that, I'd like to acknowledge we have two city council members here, TJ Hunterlaw and Kathy Grossman are in attendance. And there's actually events throughout this week as part of the city's collab series, and they're going to be helping us throughout the week with that. With that, I'd like to introduce one of our special friends, the CEO of Myriad Neuroscience, Mark Verratti, has been one of our partners and friends and also helping to bring this to the City of Mason. So with that, Mark.
Good morning, everyone. All right, so the challenge for all of you today is you're going to hear fantastic speakers today, right? And you're going to go home and you're going to tell all of your parents about it, and they're going to say, how did that happen? And most of you are going to go to your phones and you're going to try to Google it and say, how did Dr. Neil Lamb end up here today? And I'm going to give you a little sense of that because you're not going to find it on Google. How that happens is because all of you happen to live in an unbelievable city. You live in the city of Mason. And Mason works with the schools. They work with folks like me, who then find folks like Dr. Neil Lamb to make it all happen. And so I know all of you know that things just don't happen organically. They just don't happen all of a sudden. So today, I want you to think about, first of all, you're going to focus on all the presentations today, but tonight when you go home, I just want you to remember that it all started because you live in the city of Mason, who makes all of those connections. And somewhere down the road, it's going to make a lot more sense than today, especially on Monday, the day after prom, and early at 8 o'clock in the morning. So on that note, I want to turn it over to why all of you are here, which is to listen to Dr. Neil Lamb. How many of you saw Dr. Neil Lamb last year? Anyone understand? A little bit. How many of you worked with the Chromo socks? A little bit. No one? How many are excited to hear Dr. Neil Lamb today? Somebody? There we go. I had to warm them up for you a little bit. So on that note, I'm going to bring out Dr. Neil Lamb 2.0 back for a second tour of Mason. He's in the back. He's coming. Here he comes, the man of the hour, Dr. Neil Lamb.
Good morning, everybody. All right, so let me come down on the floor. All right, let's try this again. Good morning, everybody. Good morning. Excellent. So, how was your weekend? Pretty fun. Some of you were at prom, and the after prom, which just looks like incredibly epic, amazing. Very impressive, very impressive. So it's a real honor for me to be with you guys. And what we're going to do is we're going to talk a little bit about new findings in the field of genetics and genomics. We're going to cover a lot of ground. We're going to cover it relatively quickly, but I also want to make time for questions. So if there's something that you see that you want to know more about, throw up your hand, we'll stop and we'll dig into it as time permits until they drag me off the stage because you guys have another class to go to. So let's just start with some basics. It is almost impossible nowadays to look at the news without seeing some new finding that relates to the field of genetics and genomics. And so we're going to hit just a few of those this morning, but let's start with some basic. Your genome. So your genome is the collection of all the DNA that is in one of your cells or multiple of your cells. You have actually 2 copies of your genome, the one that you inherit from your mom that came in the egg and the one that you inherit from your dad that came in the sperm. So if we just look at one of those 2 copies, there are this many nucleotides. That's the building block. Those are the A's, T's, C's, and G's inside your genome. So more than 3 billion. That is an enormous amount of information, and about 21,000 genes. Those are the recipes that tell your body how to make the specific proteins or the other products that your body needs. If we look at both copies, the copy you got from mom and the copy that you got from dad, there's about 23 million places that you are different from the person sitting next to you, or the person at the high school on the other side of the city, or the person that comes from a country on the other side of the globe. So 23 million differences. That comes out to a little bit more than 1/100th of a percent difference between you and the person next to you. So here's an example of that. So here's a container of beads. I've got 1,000 beads in here. And I've got 1 bead. I don't even know if you can see it in the front row. You might be able to see it. The yellow bead. That is the 1/10 of the 1/100th of 1% that you differ between any 2 people. So this whole concept of, yes, we are all unique and we are all different and we are all snowflakes. Anyway, the reality is at the genetic level, we are almost identical to the people around us. And if you look at somebody and the things that we often classify difference on, hair color, eye color, the amount of melanin pigment that's in your skin, that's about maybe 15 genes out of 21,000 genes. So takeaway message, yes, we are different. Those differences are important, but we are so much more similar than we are different. Let's see, you guys are 16, 17, 18, yes? Okay, so when you were born, or right before you were born, it cost about $750 million to sequence your genome, to go from start to finish, to read through all 3 billion of those letters. That is not pocket change. Most of us do not have that lying around in the cushions of our sofa. It was done in a research study, and that was it. And the question was, so now we spent all this money, what are we gonna do? Where's this gonna go? The technology has changed so rapidly that today I could sequence your genome for a little more than $1,000. And I could do it with 1 research technician in a weekend. So that's a game-changing kind of approach. That means you are gonna step into a world where having DNA is a given. Having access to genetic information is a fact. Whether that's your own genetic information or the genetic information of the crops that we plant out in the fields or of the animals that we see out in the wild or at the zoo, we now live in a world that is driven by or influenced by genetic information. Let's talk real quick to lay some groundwork about different types of genetic tests. Okay, so here's your genome, and in fact what we've got is, here are images of every single one of your chromosomes. Chromosome 1, which is the largest, 21 and 22 are the smallest, and then this is an individual that's a male, they've got an X and a Y. Ladies, you have 2 copies of the X chromosome. Gentlemen, you have an X and a Y. That $1,000 to sequence your genome gets you about 85% of your DNA fully informed. So I can read about 85% of all of the information in your genome if I'm gonna sequence your genome. Now, I said just $1,000, it's actually more like 5 because somebody has to take all the data and analyze it. So if you just had your genome sequenced and said, just give me the raw data, someone would hand you a 1 terabyte hard drive and say, okay, have at it, good luck. And the next panel that we're going to talk about, the first panel this morning, we're going to talk about the influence or the connection between life science and data science, because the field of informatics is enormous in this arena. If you like computers, if you like coding, and you also like biology, your ticket is written for you should you choose to step into the field of informatics in genomics. But if right now, most of us wouldn't know what to do with a terabyte drive. So somebody else has to analyze it and tell you the important pieces. And that's why it's really more like $5,000, not $1,000. But having your whole genome sequenced gives you about 85% coverage. You could also go to a direct-to-consumer site like 23andMe. Some of you may have heard of 23andMe. You swab the inside of your cheek, or you spit in a tube, and you send that off to this company in California, and they actually don't read your genome. They don't look at all 3 billion letters. They look at about 1 million specific sites, and I just kind of cartoon-wise showed you this with these red arrows. They're looking at about 1 million sites all across your genome, many of which we know are important, and then they're pulling information based on what's in those 1 million sites. It's a lot of content, but it's 1 million sites versus 3 billion bits of genomic information. So going to a place like 23andMe or another direct-to-consumer site is nowhere near as robust, nowhere near as comprehensive as having your genome sequenced, but it's about $100 compared to $5,000. That's an easier trade-off. So that makes it more accessible, but you got to know the caveats around what you end up with. And then there's a whole nother set of tests that are more targeted, that are, sometimes they're called a panel. This is, I'm looking for a specific set of tests. Maybe you have somebody in your family who's been affected by cancer. This is used a lot in cancer. They take the tumor and they analyze the tumor for specific genes that they believe are involved in the formation of the tumor, or that they think may hold the key to how you actually treat the patient. So this is a targeted panel. This can be anywhere from a dozen, maybe $25 to $50, all the way up to several $100s. But it's much more targeted. So your world. And you probably get really tired of people saying, when you grow up in your world, and I just did that to you, so I apologize. But the environment, the ecosystem that you are stepping into is going to be an ecosystem where it is going to be assumed that you have access to your genetic information. And what do you want to do with that? Do you want access to your genetic information? It is your data. There are lots of different views on this. My particular view is it's your data, you should have access to it, but you should make sure that somebody gives you the caveats around what does this mean, and the challenges around how do you not interpret it in the wrong way. And you can take it to your physician. And I work really closely with physicians. I taught medical school when I was at Emory for a dozen years. Physicians are still grappling with how to incorporate this into your healthcare as well. You've already heard, yes, ask away. How accurate is the information from 23andMe? Yeah, so that's a great question. So I did 23andMe, and I've got my 23andMe results. And again, recognize it's a tiny fraction of information. And it's based on the science that we know at the time. So for example, my 23andMe results say it's very unlikely that I will experience balding. Well, as you can see, that is completely wrong. My 23andMe results also tell me that I am at a higher risk of restless leg syndrome. Well, that actually runs in my family, so that kind of makes sense. 23andMe also offers testing now that gives you a risk of breast cancer likelihood, if you have some specific mutations in breast cancer. They look at 3 mutations that are associated with breast cancer in one specific population. They are not looking across the spectrum of all the genes involved in breast cancer. And there are examples of women that were told that they didn't have a mutation, and so they chose to forego their health screenings, but it actually turned out they had a mutation that put them at an 80% lifetime risk of cancer. but it was not one of the mutations that 23andMe tested. So I think the important take home is it's valuable information, but you've got to realize it is a fraction of the total information. And it's based on the science at the time, and the science continually changes. So would I say don't ever do it? No, I would never say that, but I would say take it with a really large grain of salt. Maybe like a pound of salt. All right, so one of the places that you are most likely, you as a consumer, to intersect with genomics is in the field of pharmacogenomics, which is how does your body break down drugs? If any of you have ever taken a medication that did not work for you, or worse, a medication that actually made you sicker or had all kinds of side effects, you can blame your DNA. Actually, you can blame your parents because they gave you your DNA. But your genes break down these medications, turn inactive drugs to active drugs, and then break them down even further into inactive components. And your genetics controls if certain medicines work for you or don't. And right now, we treat everyone almost as the same, and we give them the same medication and the same starter dose, even though we know that that doesn't work for all sorts of people. The hope is that you actually can use genomic information and be able to categorize people based on this medicine is more likely to work for you. This medicine would work, but at a different dose. You should actually try a completely different medicine because the standard medicine isn't likely to be effective. And you guys are really lucky because in your backyard, you've got Myriad Neuroscience. And that's exactly one of the key things that they focus on, is trying to identify what medicines work and what medicines don't. So before you ever even take a medication, you've got a sense of, you and your doctor have a sense of what you should consider and what you shouldn't. So that's a test that actually involves you and your doctor. These direct-to-consumer tests like Ancestry.me discover a more complete story of you. Doesn't that sound nice? A more complete story of you, all based on your DNA. Find out where you came from, who your background is. Millions of people have uncovered something new about themselves. You can too. And then 23andMe, which we just talked about, which does ancestry. It also does some health issues. And it also does some really interesting things like when you eat asparagus and then you go to the bathroom, can you smell asparagus in your urine? So some of you are now thinking back and going, oh yeah. And some of you are like, okay, that's totally disgusting. I can't believe he just went down that road. But that's controlled by your genetics. It's called asparagus anosmia. Can you smell the asparagus breakdown in your urine after you've eaten it? Let's talk about some other direct-to-consumer testing that I would recommend that you not pay attention to, even though you're gonna get lots, you're gonna get bombarded by, hey, your DNA can tell you all sorts of stuff. So this is a whole bunch of stuff about what workout plan should you choose and what diet should you choose. Should you be on a paleo diet, a keto diet, a high protein diet, a low carb diet, Atkins, South Beach? Should you be doing interval training, workouts? Should you be doing high intensity, multiple reps? Should you do Pilates, yoga, any of those pieces? Your genetics is not going to give you that information. But there are lots of companies that would really like to sell you a genetic test. Oh, and by the way, maybe some nutritional supplements that you should also take based on your genetics. I think you want to be really careful about that. In fact, here's one of my favorites. I'll let you take a look at this a second. DNA soccer. Does your DNA give you the right information to say that you should play soccer? It probably always does. But then right here on their website, it says it's your training that matters. So why are we even taking a genetic test if it's your training that matters? So there's a kernel of reality in this, just like most good urban myths. The kernel of reality is that there are some genetic factors that give us a sense of if you have fast twitch or slow twitch muscles. So are you more likely to excel in speed, in bursts of speed, or in endurance? In other words, should you be a sprinter or a marathoner? Now, that's the kernel of truth in there. The reality is, it's the thousands of hours that you put into practice, whatever it is that you do, that drives how well you're gonna excel in soccer or softball or football or whatever, way more than what your genetic information is. So Soccer genomics, the DNA soccer test, I think you should pass on that. I would not be asking for that for Christmas, birthday, or graduation gifts. Here's another great one. And again, these are direct to consumer, so there's no physician involved. You order the test, you spit in a tube, and the results come back directly to you. These are all about your health and your longevity. How old will you be? Any of you guys heard of telomeres? Okay, so telomeres are, they're at the ends of your chromosomes. They protect your chromosomes. They're kind of like aglets. Some of you guys are at the right age that if you ever watched Phineas and Ferb, you remember the aglet song? Yeah. So I will not sing that for you. But telomeres are at the end of your chromosomes, and they protect your chromosomes. They shorten every time your cells divide. Your telomeres get a little bit shorter. And as you get older, you can measure the length of your telomeres as a rough guess for how your cells are performing. Are your cells behaving as if they're old, or are your cells behaving as if they're young? Fun fact, when astronauts go up in space, their telomeres actually get longer. And then when they come back to Earth, their telomeres get shorter again. So, telomeres are a really, really rough approximation. I wouldn't be using them to determine, when it's time to take out that life insurance policy. But there are plenty of companies that would very happily sell you a telomere test to determine the health of your cells. And then I love this one over here, My DNA Age. Are you biologically younger, or are you biologically older? And those of you that are in graphic design and arts are quick to notice the visuals that they have used for this individual to differentiate older and younger, implying all sorts of things. It's more desirable to be younger and to be beautifully made up as opposed to being older with saggy skin and thinning hair. It just feeds right into that whole culture. Again, nothing to this. This is a waste of your money. Almost as much as this. Yeah. So now it's swipe left or swipe right based if you're a genetic match. No. There are so many things that determine if you are compatible that have nothing at all to do with DNA. Although there is one relatively new DNA finding that actually seems to affect males more so than females, and it impacts a man's ability to recognize when his partner is giving him supportive social cues. So in a conversation, when a couple are talking about a problem, about an issue, if the spouse is using nonverbal cues and is being supportive, there's a specific genetic change that for some men who have this change, they are oblivious to the fact that their partner is actually supporting them. And they feel that their partner hasn't heard them, and they rate their relationship as being less satisfying. Some of you in this room and some of you over here are going, oh, okay, well that now makes sense. But again, should you build a relationship based on genetic compatibility? No, we should not go down that road. Do not go down this road. Just don't. All right, let's look at this one a second. Child development by a company called Origin. So this is for a parent that can have their child's DNA sample sent off, and then they'll come back, the results come back with this sort of information about your child's development. Okay, let's just do a straw poll. How many of you would want to know this information about your child? Let's all project forward. You've got children. How many of you would want to know this information about your child? All right. How many of you would not want to know this information about your child? Okay. And a handful of you are maybe not sure, or maybe it's early Monday morning. Fair enough. So there is some real value in knowing information about your child as it relates to their health, if you can prevent the development of disease. There's a lot of value in that. In some of these other things, there might also be value. There might be a possible reason why you would want to know some of this information. But again, remember I told you fitness and nutrition and even behavior, we know we know so little about the genetics that this is really the equivalent of me giving you a 1,000-piece puzzle and taking away 900 pieces and then saying, okay, what's the final piece look like? What's the final completed puzzle? What's the image on the completed puzzle? But I've taken away 90% of your information. Right now, we don't have a lot of information. There are some circumstances where it may make sense to do this, but right now you still probably want to do this in conjunction with a physician, not just, hey, I just want to do this. I want to do this test. I know of individuals, of stories of parents who wanted to have this information done because they were, for example, on a limited budget and they had multiple kids and they only had enough money to send one kid to college and they wanted to use a genetic test to determine which child they should send to college and which ones they shouldn't. Yeah. So, clearly, your success in school, yeah, there's a little bit of genetics to that, but there's so much more than genetics, so much more than what you inherited that drives that. And so making some of these decisions, especially on a child who can't say, I'd rather not have this information. That's kind of challenging. If your parents have gotten all of this information on you, that's taken away your right to say, I'd rather not know this information, or I'd rather wait until I'm older to know this information. All right, let's shift gears. Let's talk about forensics a little bit. Anybody in a forensics class in here? Does Mason have a forensics class? Okay, cool. So we talk about DNA testing. You watch crime scene television shows and they take a DNA sample and within 20 minutes they've got an answer and we've got our suspect. So it's a little more complicated than that, but essentially the concept is we are looking at about 20 regions across the genome in typical forensic studies, and we're comparing the genetic variation that we find at the crime scene with a potential list of suspects. Or maybe we're trying to identify an unknown body, and so we're comparing it to a list of potential individuals that it could be, and then we're looking for a match. And we can exclude some individuals and we can't exclude others. That's what we've done for forensics and DNA and criminology for probably a couple decades now. The field is really rapidly changing. And now we're moving into something that's called forensic DNA phenotyping, which means, by the way, those 20 sites that they were looking at had nothing at all to do with your genetic recipes. They were uninformative for health or physical traits. They just served as identification purposes. Now we're beginning to shift into a different approach as we learn more about the genetics that controls things like hair color and eye color and skin tone and facial shape. So we're now beginning to be able to say, I can use genetic information and paint a picture of the individual that left this DNA sample behind. Now, it's important to note that there's a lot of these pieces that we don't fully have a strong genetic picture of. We're still learning, but we've got enough information that there are now companies out here that can take a genetic sample and use computational modeling and informatics, and they can predict the face of the individual that that sample belongs to. So again, it's predictive. It's an estimate. It's not saying this is exactly what they look like, and you can change it based on, you can age the individual, you can gain or lose weight. So there are all these new ways that you can now begin to identify samples. And there's a company called Parabon Nanolabs that now does this. They work with cold cases with police from around the country, and they actually take this DNA sample and they run it through their database, and they actually come up with a predicted profile. And in about 50 cases, that predicted profile has actually led to an arrest and a confession and a conviction. So we're now beginning to get to the point where we can start to say something about the individual that left the sample behind. Let me take that in a couple weird directions for you. And maybe one that's not so weird. The other thing that we can do with that data is we can now compare an individual's sample with all of the ancestry, the genealogy databases that are out there. So about a year ago, in fact, a year ago next week, police arrested this gentleman in California and charged him as the Golden State Killer. The Golden State Killer raped and murdered dozens of women throughout California in the 70s and 80s. Was never caught, and then the crime spree stopped. Police had DNA samples, but the DNA didn't match anyone in the known database, so they couldn't go anywhere with it, because it only works if you can make a match to somebody that's already in the database. So a group of forensic detectives did something a little unique. They actually took the DNA sample and they sent it off to a company, and they got an ancestry profile on the DNA sample that was left at the crime scenes. And then they went to open access databases. So they didn't go to like Ancestry.com or 23andMe, because those sites don't give up their information, but there are plenty of websites that people can just randomly, freely dump their data in and say, help me find my relatives. And so they put the data in, and it led them to this guy's brother. And then they followed this guy who used to be a policeman, and they waited until he went into the grocery store, and they swabbed his car handle, the car door handle, where his DNA was on his fingerprints, and they made a match. And so there have been probably 36 cases that have come out like this, of using your ancestry to actually identify somebody in your family. You know, is that something that we want? On the one hand, it's public safety. We're taking a criminal off the streets. On the other hand, is that what you want your genetic information used for? An artist in Texas actually took people's food samples in Central Park. So I would eat like half a hot dog and throw it away, throw it on the ground. This artist would swab the bite marks and use this same technology and build a 3D mask of the people that had left the food that had littered. Yeah, and then hung it up in an art gallery in New York. And then, here's an interesting concept as well. Could you conceivably, now this is not real yet, you can do this by taking photos of an individual, building a 3D model of their head, and then using that as face recognition to unlock their phone with a 3D model. But could you use your DNA to build a facial recognition, build a 3D model, and then unlock your phone? That's a lot of work to get into somebody's phone. But it is that stuff of science fiction. All right, let me wrap up with a couple of really amazing health stories, specifically for individuals that deal with cancer. Traditional therapies, chemotherapy, just kills cancer cells. It's like a sledgehammer. This really cool new field of immunotherapy, if you know individuals with cancer, there's a fair chance that a high percentage of them, when standard therapies fail, they may be offered immunotherapy. And immunotherapy is almost seems too good to be true. But here's the concept. Can I selectively awake your immune system to recognize your cancer and to attack and to kill your cancer? So here's an example. These are 2 new drugs that have been approved by the FDA. They're called CAR-T therapies. One is Kymera and one is Yescarta. Kymera works for leukemia and Yescarta works for lymphoma. And this is the science behind that. They take your T cells. So these are your immune cells that help trigger your immune system. They take the T cells out of your body. You give a blood sample. And then they genetically, this sounds like science fiction, they genetically re-engineer your T cells. So your T cells now recognize and hone in on a specific protein that is uniquely found in your cancer. So the scientists have first looked at your tumor and figured out what is unique about your tumor, and then they've reworked your immune cells to specifically go after that cancer and attack that cancer. So they do all this in the lab, they grow it up, and then they transfuse it back into your body, and these reworked immune cells attack your cancer. You can see the price tag, because every time you create this, you have to create it uniquely new for every single patient. So it's called a living drug. But for the patients that have gone through this, in more than 3/4 of the cases, it completely obliterates the cancer. And these are patients that have gone through therapy after therapy with often no luck. So this whole concept of immunotherapy and targeted drugs using the genetic information in your tumor, this is changing the way that we think about healthcare. And then let me wrap with this. You are stepping into a world where genomic information is now going to be used to think about treating diseases in completely new ways. Last year, a woman who was pregnant with twins that had a specific rare genetic disorder, physicians gave her fetuses a specific injection of the protein that they were missing that was caught from their genetic disorder. So they were in utero injected with this protein, and it led to what looks like completely typical development. So these kids, even though genetically they've got the mutation, because they got this protein in utero, they won't have any of the symptoms of the disease. And we're looking at gene therapy as a treatment for sickle cell anemia, for multiple forms of inherited blindness. We use gene therapy to actually, for a child whose skin would shed off, it would just slough off under pressure. So clinicians genetically re-engineered his skin. They took out the mutation, added in the correct gene, and gave his skin grafts over 80% of his body. And the kid now plays soccer and is rough and tumble and doesn't have any trouble anymore. And then this guy here on the right, this is Brian Maddox. Brian Maddox has a genetic disorder that causes him not to produce a certain protein that stunts his growth and damages his organs and ultimately leads to mental impairment and death. Last November, scientists actually transfused him with something called gene editing. You guys use Microsoft Word or Google Docs, and you know what find and replace looks like. Gene editing is the genetic equivalent of find and replace. Find this mutation and edit it, change it to this. So scientists gave him a transfusion that specifically went to his liver and edited a subset of his cells to change the mutation to the correct copy. It's too early to know how effective that is. It's going to take a while to figure that out, and he's the first in a long list of patients. But you are now on the cusp of a world that will be shaped by genetics. From the medicines you take, to the food that you eat, to the tests that you are offered at the doctor's office, to the tests that you may be offered about your kids, to the way we think about treatment and therapy. It is all shaped and controlled by genetics. That doesn't mean that it's all the best thing since sliced bread. There are lots of ethical questions that we have to step into, and you guys have to be the ones that actually have those conversations and those discussions. If you like the field of science, if you like genetics and genomics, and if you're thinking about healthcare, if you're thinking about physical therapy, or you're thinking about nursing or about PT school, this is going to be at your doorstep. If you like physical therapy, PT school for physical therapy, sorry. If you like informatics, again, I said you can write your ticket here. This is the place for that. Okay, I think I have like 3 minutes. So let me see if you've got additional questions. We're going to do an Ask Me Anything in our session in a little while, but questions about any of the stuff that we've just talked about. It is Monday after prom. I would be that exact same way. Yes?
Do you think immunotherapy will be able to be generalized to all cancer?
Ooh, do I think immunotherapy will be generalized to all cancer? No, because not all cancers has the immune system gone blind to. So I think in a number of cases, immunotherapy will be valuable, but I don't think it's going to be the magic bullet for everything. And for a number of patients, immunotherapy doesn't even work. So they spend hundreds of thousands of dollars and they don't get any results. Anything else? Yes, and then we'll go right back here.
So if you have to make a unique immunotherapy for every patient, will you eventually run out of ways to make
Will you run out of ways to make it if you have to make a unique one? Fortunately, no, because essentially the unique thing you're doing is you are looking specifically at the proteins that are present on the surface of the cancer cell, and then you are building a drug that specifically attacks that. So it isn't that there's only a limited amount that you can work with. Was that the bell? Is that what that sound is? Okay. All right. Thanks, guys. Have a great day. Good to see you.